Hello guys, welcome back to our revision series, and this is biology paper 2 Kcse 2019. Now let's move to our first question. That is in section A. In an experiment to investigate the effect of sodium chloride on the growth rate in a spinach seedling, seeds were treated with different concentrations of sodium chloride. The results are recorded in the table below. As you can see, in the first column, we have concentration of sodium chloride in moles per liter, 0.00, 0.06, 0.12, 0.18. You can see it is increasing. Then percentage of spinach seeds which started to grow roots, 99.98, 98.20, 92.0, 54.0. You can see the percentage which started to grow roots are decreasing with increase in concentration of sodium chloride. Then mean root length 17.70, 15.60, 10.20, 7.60. As you also can see, the mean root length is decreasing when the concentration of sodium chloride is being increased. And before we look at part A, you need to know in paper 2, the geography of paper 2, one question must have a table, and this table, so the graph helps us interpret what is in that table. So the students must be very keen when answering these questions on the tables, must be able to tabulate the results and able to understand how the components given are increasing or decreasing and the reasons for that. Part A, from the results in the table above, explain the effect of increasing concentration of sodium chloride. And increasing concentration of sodium chloride decreases water potential, or in other terms we call it osmotic potential, or you can say increases osmotic pressure, and in other terms or simple times you can say the seedling in the surrounding solution becomes hypertonic to the cells up in the seedling, and the seeds or the seedlings take in water by osmosis, and they become dehydrated or lose water molecules to the surrounding solution, reducing the growth activity, hence reduced growth rates. That is how you should explain. Then part B, apart from a ruler, state two other equipments one will need to determine the rate of growth in the roots. And you can have a thread or a string, a book, which you can draw and be able to obtain the length and calculate the rate of growth. Then we have a marker pen for marking those roots. Then you can have dyes or waterproof ink, blotting paper. Then part C, with a reason, state one other part of the seedling the students will focus on to determine the effect of sodium chloride on the growth, and that is the rate of growth or increase in length of the shoot tip of the apex. And this is because it is also a region of cell division or growth in plants. Part D, set the likely effect on the seedling of increasing the concentration of sodium chloride to 2.20 moles per liter, and this will cause the seedling to be dehydrated or lose water, hence dry or die or wither. Then question 2, the table below shows results of blood cell counts per millimeter cubic of blood from a sample of people living at different altitudes We have red blood cells, white blood cells, and the altitude. As you can see, the altitude is increasing up to 4500, and the white blood cells count is a constant 0.5 across, while the red blood cells count is increasing 4.8 to 9.82. That is when the altitude is increasing, the red blood cells increases. Explain the relationship between red blood cells count and altitude. And the red blood cells, or the number of red blood cells, as you can see here, it is increasing with the increase in altitude, and this is to increase oxygen carrying capacity, since oxygen concentration is lower at higher altitudes. Then Roman 2, white blood cells count and the altitude. And the white blood cells have to protect the body against harmful microorganisms from the pathogens, and the quantities of pathogens in the body microbial attack is not dependent on the altitude. Hence, the number of white blood cells count is a constant at whatever altitude. In short, we are saying the white blood cells count remains constant because it is not affected by altitude. What affects the white blood cells count is the presence of pathogens in the body. Then part B, explain why chances of nose bleeding increases with altitude in humans, and that is atmospheric pressure decreases with the increase in altitude, and this imbalance between the outer atmospheric pressure at high altitude and the internal blood pressure generated by the heart results in one, nose bleeding at high altitudes, stroke internal blood pressure is higher than the outer atmospheric pressure. That results into one, nose bleeding. Question 3, part A, state one importance of irritability to living organisms. And basically, irritability means responding to changes in the environment or sensitivity, and this enables living organisms to avoid dangerous or harmful or harsh climatic and environmental conditions. Avoid must be there, or dangerous must be there. It also enables living organisms obtain or access favorable conditions, e.g. light, water, oxygen, mates. Favorable must be there, and obtain also must be there. Part B, in an experiment, students treated seedlings as illustrated below. We have unidirectional source of light, Roman 1, Roman 2, Roman 3 seedlings. Roman 1 seedling is unaffected at the tip. Then Roman 2, it is decapitated. It means the shoot tip has been removed. Then the third one, the tip is covered by an opaque cap. And in Roman 1, account for the observations made in seedling 1. And that is a positive phototropism. The tip bends towards light, and the light causes migration of auxins produced at the tip to the darker side of the shoot. And this results into faster must be there, faster division of cells, or elongation or growth on the darker or opposite side of the shoot, hence bending towards the light. It means when exposed to this unidirectional light, the auxins move to this darker side, or migrate to the darker side, and we will have a higher concentration on this darker side than on the lit side. Hence, faster cell elongation or cell division on this darker side than the lit side, causing the shoot to bend towards the source of light. Roman 2. Explain the similarity in the end results made in seedlings 2 and 3. Let's look at 2 We have 2 here, and 3. 2 which was cut at the shoot tip, and 3 the tip covered by an opaque cap. Both seedlings remain upright. We can accept rect. Seedling 2 does not have the tip, while 3 the tip has been covered by an opaque material, thus preventing light, causing an equal distribution of auxins. Or we can accept uniform or even distribution of auxins. Then Roman 3 said the likely treatment that will make seedlings 2 and 3 respond like seedling 1. The 2 seedling Roman 2 can fit an agar block treated with auxins, or can say the IAA, which is indole acetic acid, which is an auxin, at the decapitated end of the seedling. So in short, we'll place the agar block here, which is treated with auxin or indole acetic acid, in order for it to give us the result obtained in this seedling 1, which is bending towards light. Then Roman 3, seedling Roman 3, you remove the opaque material covering the tip of the seedling 3, or replacing the opaque cap with a transparent one. Next question, question 4. In cats, gene for fur color is sex-linked. It means the male have the the male have the XY, female have the XX chromosome. Letter G represents the gene for ginger fur color, while B represents the gene for black fur color in a given cat species. And these genes are co-dominant. Heterozygous females have ginger and black patches of fur, and their phenotype is described as a tortoise shell. With reference to the information above, what is meant by the term co-dominance? And co-dominance is a condition where both alleles are expressed or shown equally in the phenotype of the offspring, or none suppresses the other gene for ginger and black fur color from expressing itself in the phenotype. In short, the alleles are expressed equally in the phenotype of the offspring. Because you can see here, the heterozygous females have ginger and black patches of fur, and their phenotype is described as tortoise shell. And I was saying we have one parent having a ginger fur color, and the other one has a black fur color. The offspring will have ginger and black patches. So that is why in this case we're saying that codominance alleles, both alleles are shown equally in the phenotype, and the phenotype is saying it is the outward appearance of an organism, and that is of the offspring. It means the offspring will have both ginger and black patches, being described as a tortoise shell. Part B, explain why male cats with a tortoise shell phenotype do not usually occur. And the gene for coat color is contained on the X chromosome. Contained on the X chromosome. Males only inherit one X chromosome from the females or mothers, or in other terms, Y chromosome does not carry the gene for color coat. That is why we don't have male cats with a tortoise shell phenotype. Remember we talked about this gene for fur color, which it is a sex-linked. Part C, a tortoise shell female was crossed with a black male, determine the genotypes and phenotypes of the offspring. 5 marks. In order for you to get the 5 marks, you must cross correctly, and we have parental phenotypes: tortoise fur female and a black fur male, which are the parents. The genotypes will be BG for the tortoise fur female, because these two are linked on the X chromosome, and because it is sex linked, you must have an XX on the female, then superscript capital B, capital G, then you cross with a black fur male, that is X superscript B, then Y is not associated with this gene for coat color. This X or cross must be there, that the gametes should be encircled properly, then these crosses should not pierce through the gametes. Then the results are here. We'll have the genotypes. We have a female offspring which has a black fur, because we have both here, which is a homozygous, capital BB. Then a female with a tortoise fur containing both black patches and ginger color. Then we have the male black fur, XY, the X superscript to B, then a male with a ginger fur color, capital G, then we have the Y without anything. You can use a Punnett square, and the Punnett square, this cross here separating these symbols, this one is a male, and when you put this symbol you must be very keen, because this male will have the X, Y this side, then because this symbol is for female, have the XX on this side. Must be keen, because most students don't remember or confuse the two. You get the same results. Question 5. A person accidentally touches a hot pan and responds as illustrated in the diagram below. We have the hot pan, forearm withdrawn, then we have the spinal cord. Part A. Explain how the response illustrated above occurs. 6 marks. The stimulus, which is the heat or pain, is detected by temperature or pain receptors. Receptors must be there. We can use also the thermoreceptors in the skin. An impulse is then generated. The impulse is transmitted along a sensory neuron to the spinal cord or the central nervous system. Then transmission across the synapse to relay neuron, then across a synapse to motor neuron, across a synapse to the biceps muscles or effector organs via motor neuron. Then the muscles or the effectors contract and the hand moves away or is withdrawn. Is known as simple reflex action. And this question, basically you need to know the reflex arc, and the reflex arc is here. The pain receptors detect the pain, an impulse is generated, the impulse is transmitted to the central nervous system through the sensory neuron, then via synapse. A synapse is the junction between two neurons, to the relay neuron. Relay neuron, there is interpretation, the central nervous system, then via synapse, the nerve impulse is transmitted to the motor neuron. Motor neuron via synapse to the bicep muscles which contract, and the hand is withdrawn. That is how you answer such a question. Part B. Explain how auxins are utilized as selective weed killers in agriculture. And selective weed killers contain auxins which are absorbed by the weeds, making the weeds to grow abnormally or die out ahead of the beneficial plant. That is how the auxins are utilized in selective weed killers. That is a part A for our revision. Please like, subscribe, share, comment. We shall look at part B later on, and don't forget to come back and look at part B. Thank you.